Hello, I'm Phil, and I'm going to introduce to you a web-based Shiny application which can be used for population genetics analyses required in forest genetic monitoring within the LIFEGENMON project. There are numerous standalone software applications available to perform population genetics analyses. These programs can be multi-purpose, carrying out a wide variety of calculations and statistics, such as the FStats program, which can be used to calculate most basic diversity indices, F-statistics, Hardy-Weinberg equilibrium tests, and more. These programs can also be tailored to specific research questions, such as the Migrate program and Structure program, which can be used to assess the effective population size and optimum number of genetic clusters, respectively. Within a typical analysis pipeline, you might use several of these programs, demonstrated here by the image on the slide showing a selection of the programs available and how data moves between them. This can be a very time-consuming process to put this pipeline together, and it's complicated by the fact that many of these programs require different input formats of the data and can use slightly varying mathematical frameworks underpinning their calculations. These programs also suffer from the fact that many of them are point-and-click applications with very little information as to what is happening in the back end of the program. This can cause issues for the reproducibility of the science conducted using them if the steps are not thoroughly and well documented, and particularly if the researcher carrying out the analyses doesn't have a firm grasp on exactly what is being done within the program. So in recent years, many of the functions of these standalone programs have been able to either be complemented by or entirely re-implemented within the R environment. This can both speed up the analysis and, very importantly, create very well documented pipeline scripts which can be used to entirely reproduce the outputs of analysis or used as a jumping off point for further analyses. Despite this, the adoption of R-based pipelines has been quite slow within the population genetics community, and also does not necessarily fix many of the problems which can be associated with standalone applications. For instance, there are still multiple R packages which can be used to carry out analyses with different mathematical interpretations, different functions, data input formats, and even data storage formats once your data is inputted. Beyond this, as a newly developing area, it may be hard to find the package which can carry out the specific analysis that you wish to conduct, if that package exists in the R environment at all. And further to this, when you're working with an R package, you are essentially provided with a set of functions and scripts, which you then have to put together to create the analysis pipeline. Again, this can be an incredibly daunting experience and almost entirely inaccessible for someone who doesn't have prior R coding experience. This is where the population genetics analysis application that we've developed for use in the LIFEGENMON project comes in. We've created a simplified, clear pipeline almost entirely within the R environment through the aggregation of several of these packages, their functions, and unique scripts that we've written. Through this pipeline, you can carry out many of the basic and fundamental analyses found within population genetics studies in a way that is accessible to the general user. While as a Shiny app, this may also seem like it's just a click and get the data kind of application, all of the underlying R script to anything carried out within it is available for download, so it can also be used as a teaching tool for R scripting in population genetics and as a jumping off point for further analyses. So now I'm going to give you a quick overview of what we do within the Shiny application and then go into greater detail of each part. So this application can be split into 5 discrete sections. These are data import, data filtering, the analysis of genetic variation, the analysis of genetic structure, and ordinations and visualization of the data. All of these sections, the processes within them are performed on a single R object called a GenInd object. This contains all of the loci and allele frequency data of each individual within your file and the associated metadata with them. So when you're importing data, the application can handle 2 different data import formats. These are GenAlex formatted and structure formatted files, which are 2 very common file formats within population genetics. After importing your file, you can firstly view the population designation of each individual, along with a print of the file that you've just imported, which you can use for quality checking to ensure that everything that you've done is fine. After this, you can then view the print of the genin file which has been created by the application, again as a quality assurance check that this file has been created. So you can keep in line with the data every step of the way. When importing a GenAlex file, this is relatively simple, as you've seen from the GIF on the right. But when importing a structure file, you have to give it a couple more user-defined numbers to work with. This is the number of individuals and the number of loci used within the file to just help the underlying R script read the file, along with the rows per individual. From here, everything downstream is exactly the same. Users can interactively define the specific population parameters within their file and use this to either analyze a single variable or an interaction of variables. As you can see here, when you choose toStrata, the Germany _A variable can be split into two, and there as the country and the age class of the trees used. Next is data filtering. And within this, there are two different filtering implementations, being basic filtering and then optional additional filters. When you carry out basic filtering, all loci and samples are subjected to this when you filter the data. The first part of this is filtering monomorphic loci, which are loci which show no variation across the entire data set and therefore can't be used to segregate individuals or populations from each other genetically. You can also filter both loci and individuals by the degree of missing data within them by a user-defined threshold. Finally, within basic filtering, you can filter by minor allele frequency. This is the frequency of the second most abundant allele at a locus, and it's generally agreed that values below 1% or 5% are uninformative loci for segregating individuals and populations. The optional additional filters include filtering for linkage disequilibrium firstly. This is because many of the downstream analyses expect the data to be completely unlinked and therefore in linkage equilibrium. This is where two loci kind of move with each other, or two alleles move with each other within a data set. Secondly, you can filter by Hardy-Weinberg equilibrium. This is a bit of a rough and ready version of ensuring that potential sequencing errors are filtered from the data. And we implement this through filtering any loci which are found to be out of Hardy-Weinberg equilibrium in over 50% of the assessed populations in your study. Finally, many researchers choose to only perform analyses on neutral markers. These are genetic markers which are thought to not be undergoing any selection pressures and therefore may be more indicative of past demographic events within a population. Therefore, within this application, you can also filter non-neutral markers out of the dataset through the FST outlier method. So moving on to genetic variation, I've again split these measures into two overarching concepts, being multiplicity and diversity, with them effectively representing the level of diversity within populations and then between populations. Both of these sets of measures can be rapidly summarized at the click of the button, as you can see in the table in the top right, by both the population assignments that they originally had in the file and by the strata that you've defined as a user. You can also perform a number of other analyses within this section and generate publication quality figures, such as the rarefaction curve for allelic richness that you see in the bottom right of this slide. So, the analysis of population structure, admixture, and hybridization makes up a large portion of the application. In the application, we implement the standard approaches for assessing between population diversity through the analysis of molecular variance, or AMOVA, and pairwise population comparisons. We also utilize two methods to determine population admixture and hybridization, along with the optimum expected number of genetic clusters through structure and a mix of Snapclust and DAPC analysis. So, using analysis of molecular variance, or AMOVA, you can detect differentiation between populations based on F statistics. Through the permutational analysis of distances derived from these statistics, AMOVA can partition the variance in the dataset into within individual variance, between individuals but within population variance, and between population variance. This gives you an overview of the hierarchy of variation present in your study system, much like how in traditional ecological analyses, a Permanova analysis can tell you if your groupings are generally dissimilar or similar from one another. Populations can then be specifically compared to each other through pairwise comparisons of their distance matrices, which is also what we've used in the bottom right of this slide to generate the dendrogram of distances between populations. So moving on to structure analysis, this is the most popular method of assessing the optimum number of population clusters, genetic admixture, and hybridization between species. But it cannot easily be implemented within the R environment and therefore still relies on a standalone application. What you can do within our application is export a structure formatted file to be used in this program and then re-import the output data from this program for further interpretation. Firstly, this output data can be used to determine the optimum number of genetic clusters through the Ivano structure method, detailed in the top right figure on this slide, where the delta K value at its highest is generally regarded as the optimum number of clusters. From that, you can then visualize the probability of membership that each individual has to these clusters based on your original population hypotheses and the mean probability that each population hypothesis has to this cluster. You can also export this data for further more in-depth examination of it. So Snapclust itself is a R-based alternative to structure analysis with the same goal of finding the optimum number of population clusters in the data set and assigning probabilities of membership to each individual. This has been shown to be able to effectively assign individuals to clusters similar to or better than structure analysis and faster, and is entirely implementable within the R environment. Discriminant analysis of principal components, or DAPC, is similarly an R-based method of identifying population clusters, but it does not perform as well as Snapclust or structure analysis. What it is very good at is visualizing your Snapclust assignments of populations in a reduced dimensional space, as you can see in the GIF on the right. I'll let it go forward to it again, as you can see here. So finally, along with DAPC, there are 3 ordination techniques implemented within the application. The basic aim of all of these ordination techniques, including DAPC, is to simplify complex multivariate data sets to low dimensional space so that patterns can be easily visualized within them. And in the GIF on the right, you can see a few of the customizations that you can make to these figures when they're generated. There we go. So I hope that in the short time I've been able to demonstrate that our application can perform all of the basic population genetics analyses that you may need for forest genetic monitoring. This can be carried out rapidly, customizably, and results in journal quality outputs. It can be used both within the LifeGen project and beyond. Thank you for your time, and I'll happily take any questions now.